Everyone. In the previous part, we have gone through the active transport, the second type of mechanism of uptake mechanism of nutrients. And in this part, we are going to discuss about group translocation. This group translocation we will find only in the bacterial cells, not in the eukaryotic cells. So first of all, let's see what is a group translocation and what is the mechanism of it. So this is all the introduction that I have given you in the part 1 about diffusion and part 2 about the active transport. And now this is the third type where we are going to discuss is group translocation. What is a group translocation? The group translocation is the process in which the substrate molecule is transported into the cell in an altered form. This is very important. Remember, the substrate molecule is transported into the cell in an altered form. And like active transport system, this is also an energy dependent process. So these 2 points are very important to remember about the group translocation. One is the substrate molecule is transported into the cell in an altered form, and the second one is it is also an energy dependent process like active transport. The best known group translocation system occurring in bacteria is phosphotransferase system, which we simply represent it as PTS. What is this PTS system? How it is working? Everything I will explain you in detail. Don't bother. Let's see what are the components of this PTS system first. The PTS, what is meant by PTS? Phosphotransferase system. This PTS consists of, or the PTS component are, there are two enzymes were named as enzyme 1 and enzyme 2. Along with these two enzymes, a low molecular weight heat stable protein is also there. What is it? Heat stable protein, that is HPR. So altogether we are having the three components, two enzymes and a heat stable protein. Now this enzyme 2 is again having three subunits or domains. What are those three? Enzyme 2A, enzyme 2B and enzyme 2C. Remember, in the PTS system we are having three components in which two are enzymes, enzyme 1 and enzyme 2, and then so here enzyme 1 and enzyme 2 and a low molecular heat stable protein, that is HPR. And this enzyme 2 is again having the three subunits, that is enzyme A, 2A, enzyme 2B and enzyme 2C. So coming to their locations, I will tell you by showing the figure, then you will come to know it very well. So here is the figure of the group translocation. So this obviously I think so you know it's the plasma membrane of the bacterial cell wall. And then these are blue color one is representing the enzyme 2, where I told you enzyme 2A, 2B and 2C. And here is the enzyme 1. And here is the HPR protein. What is HPR? Heat stable protein, which is a very low molecular weight protein. Then coming to the location of these components of this PTS system. So here this is the enzyme 1 and HPR and your enzyme 2, one of the subunit, that is enzyme 2A. These are all going to be present in the cytoplasm. You may think that enzyme 2B is also present in the cytoplasm. Yes, it is also present in the cytoplasm, but it is frequently attached to enzyme 2C component. So we are going to mention that enzyme 2B is frequently attached to enzyme 2C, where this enzyme 2C subunit is embedded in the plasma membrane. So that's all about the components of this PTS system, that is phosphotransferase system. Now coming to the process, I told what is a group translocation? The process in which a substrate molecule, so this is a substrate molecule, so here I am taking the two examples, one is a glucose and another one is a mannitol. Substrate molecule is transported into the cell in an altered manner. See here outside it is in the form of mannitol. When it came into the cell it became mannitol 1P, that is mannitol 1 phosphate. That means that's how group translocation is being defined as the process in which the substrate molecule is transported into the cell in an altered form. Let's see another example. So here is a glucose and when it is going to be entered in the form of glucose 6-phosphate. You may get a question, ma'am, how this is going to be altered when entering into the cell? That's what these enzymes all are involved. So let's see that process. In group translocation, a high energy phosphate molecule is transferred from PEP. What is this PEP? Phosphoenol pyruvate is going to release the ATP or the phosphate molecule to enzyme 1. So here the phosphoenol pyruvate is being converted into the pyruvate. So this phosphate, whatever present in the phosphoenol pyruvate, that phosphate group is being transported to the enzyme 1. Now this enzyme 1 is getting excited because of having the energy and it moves that phosphate group to the HBr protein. And after transferring the phosphate to the HBr protein, the enzyme again goes into its normal state of enzyme 1. Now this HBr also gets excited of having the phosphate group, that means energized phosphate group. Now this HBr will be carried by the freely present in the cytoplasm, the component of E2, that is E2A. And now this phosphate from the HBr, that phosphate is being taken up by the enzyme 2A subunit. Once it has taken, it will move near to the E2B like this. Now it attaches to the E2B and transfers this phosphate group to the next to E2B, and from this E2B, once it gets excited, now that energy will make this mannitol to sorry, so this mannitol is being dragged or pulled inside and being converted into the form of mannitol 1 phosphate. That means the E2B, that is enzyme 2B, is transferring this phosphate to a molecule called as mannitol or the substrate and converting into the another form. So that's how this enzyme 2 is involved in the transfer of this substrate molecule into another form, that is altered form, as some mannitol 1 phosphate. So take another example. So here again the phosphoenol pyruvate is liberating the this phosphate, and that was taken by enzyme 1 and it is being transferred to the HBr, and from there it is being taken by the E2A. It will move to the E2B, and once it moves to the E2B, now the whatever the sugars, it may be a mannitol, glucose or some else, will takes this one and it will be in the form of a glucose 6 phosphate. So this sugar molecule present outside the plasma membrane is phosphorylated and is transported across the plasma membrane by the enzyme 2. So that's enzyme 2 transports only specific sugars and varies with PTS system. Okay, so I think so you have understood this whole concept of group translocation, which will be very useful in the examination as a short. So try to look after it. Okay, so what are the molecules that were taken by this group translocation? In the past two other types, that is your diffusion and your active transport, they are having their own usage of that system. In the same manner, this group translocation is used by the bacteria to take variety of carbohydrates such as here glucose, mannitol, fructose, sucrose, N-acetylmuramic acid and others by this PTS system. So what is a PTS system? Phosphotransferase system. Okay, I think so you understood all the three types of uptake of nutrient mechanisms, and that's the end of this topic, that is uptake of nutrients. Thank you.